Okay, so in today's experiment, we're going to be looking at experiment 2, which is focused on gas chromatography. Now, gas chromatography is a really powerful technique for separating mixtures of volatile compounds. So in this experiment, one of the first separations that we're going to be looking at is the separation of cyclohexanol and heptanol, shown here. Now, cyclohexanol, as we've noticed, contains an alcohol functional group. And what kind of intermolecular forces do alcohol functional groups have? What kind of intermolecular forces do alcohol functional groups have? Hydrogen bonding, right? While the aldehyde functional group, by comparison, primarily engages in dipole-dipole interactions. Wonderful. Okay, so because these two molecules have a difference in terms of their polarity and intermolecular forces, we will expect to see a difference in boiling point and a difference in how these compounds interact, engage in intermolecular interactions with other molecules. Now, as we notice from our physical properties, gas chromatography is ideal for separating mixtures of volatile compounds close in boiling point. So even though separating these two compounds via distillation, even fractional distillation, would be relatively challenging, gas chromatography is able to achieve the separation of compounds with very small differences in boiling point by utilizing their subtle differences in physical properties. So in this experiment, we're going to use preparative gas chromatography, which is a larger scale gas chromatography operation to separate a 25 microliter 1-to-1 by volume mixture of cyclohexanol and heptanol. So to talk a little bit about how preparative gas chromatography functions and what a gas chromatography instrument looks like. So we have our mixture, and let's zoom in a little bit on that. So we have our mixture, and there's an injection port. We're going to take our mixture and inject it into our port where the sample is converted into a vapor. The sample then passes through a column that is maintained at a specific temperature in our oven. Now, as our compounds pass through our column, the key feature for chromatography is that different compounds elute at different retention times based on their intermolecular interactions with the column and our stationary phase. After the compound passes through our column, it will eventually pass through a detector where we can measure and detect as our compounds exit from the column, and then we can collect each of our two mixture components in a gas collection tube. Gas chromatography separates compounds fundamentally based on their intermolecular interactions with the column and the column stationary phase and their relative boiling point. Does this make sense to everyone so far? Does this schematic make sense to everyone so far? We're going to unpack this idea, and we're going to talk a little bit more about how gas chromatography works and how the separation actually occurs in a moment. But I just want you to understand the gross schematic of a gas chromatography instrument. We have our mixture, we inject it into a port where it's converted to the vapor phase. It's carried through the column using carrier gas, and as it passes through the column, different compounds will interact with and absorb or condense onto the column surface at a differing frequency and with a differing strength of these intermolecular interactions. As a result, different compounds will elute or leave the column at different times and different retention times after injection. As a result, as we detect compounds exiting the column, we can collect fractions and we can collect each of our mixture components as they exit the column. So this is preparative gas chromatography, where we're separating a mixture of about 25 microliters of sample utilizing our gas chromatography column. Does that make sense to everyone so far? Does that make sense to everyone so far? And again, the separation is based on boiling point and the strength of intermolecular forces. So when we take a look at our detector, the primary detector is thermal conductivity. And what I want you to know from this detector is that as a sample passes through our detector, we observe a corresponding signal. And the overall intensity of the signal depends on the amount, the raw mass of sample. So we see this signal as our sample exits the column and passes through our detector as a peak, where this peak size is proportional to the compound mass. Or to more correctly phrase it, the peak size is proportional to the mass of compound. So the more compound we have exiting our column, the greater the signal that we would observe in general. So we observe a signal as the compound passes the detector. So in this case, if we have a mixture of two components, such as cyclohexanol and heptanol, we would see two peaks exiting the column at different times. So again, compounds elute through the column and pass through our column at different retention times, which are times after injection, depending on their boiling point and their intermolecular forces. Does this make sense to everyone so far? Does this make sense to everyone so far? Now, to talk a little bit about how to analyze the peaks and how to process this raw data and how to process this raw data that we've obtained from our detector into meaningful data corresponding to the mass of our sample, we're going to have to process our peaks using the tangent line method. So in your tangent line method, first and foremost, I'd like you to look at the following picture on the right. So notice how at an inflection point, we observe a change in the slope of our line. So in this case, we're going to take our peak and we're going to identify our inflection point, which is the point when our peak changes, its slope. Then we're going to draw our tangent line, which is drawn in blue in this case. And we're going to draw a line tangent to the point of inflection. From this tangent line, we can read off first the retention time, which is the amount of time required for our sample to exit the column. So the retention time is drawn from the time of injection, which is written as time is equal to 0, and the center of our peak, which is typically the point when our two tangent lines intersect. So this is representing our retention time, which is essentially the amount of time it takes for our peak to exit the column. The other piece of data that we can note, as we can clearly see for this peak does not elute at a single time. It elutes over a time range. And we can calculate the peak width. We can calculate the width of our peak by subtracting T2 from T1, which are the 2 points where our tangent lines intersect. To give us a simplest analogy for what is going on here, if we have our sample peak, so just to take our peak from scratch, so let's suppose we have a simple peak. The first thing that we are doing is we are identifying our inflection points. Which is the point when our slope changes. So we first identify our two inflection points. Then we draw our tangent lines. And we want the tangent line to only touch the inflection point. Okay, we're going to record the two time points where our tangent lines intersect the x-axis. And we're going to record the center of our peak where our two tangent lines intersect as the retention time. Based on the start and end, the first time when we start to observe our peak to elute and the time when we see our peak finishes eluting, we can calculate the peak width, which is equal to T2 minus T1. In essence, what this method is doing is it's converting our irregularly shaped peak into a more regularly shaped triangle, which we can more easily calculate the area for. So essentially, this method approximates a peak as a triangle, and then it can calculate the area of that triangle. Does everyone understand how we're processing these peaks in order to figure out the retention time and in order to calculate some additional properties and additional measurable pieces of information from these peaks that we observe as our sample passes through the detector? Does this make sense to everyone so far? Okay, so now that we've processed our peak, we can calculate the following values for our peak. First, we can calculate and read off our retention time, and we can calculate our peak width. Now, from the retention time and the peak width, we can estimate the number of theoretical plates for our column. We can estimate how efficiently our column is able to separate our compounds under the given column conditions. And the following relationship has been derived between the number of theoretical plates and the retention time and peak width. the number of theoretical plates, just like in a distillation, as we saw for a 2-plate distillate, for example, represents the efficiency of separation and the number of sort of equilibrating steps that occur as our sample passes through the column. The greater the number of theoretical plates, the greater the efficiency of our separation. Now, because columns have a variable length, because different columns will have a variable length, and because column length in general increases how efficiently we're able to separate our compounds, we want to standardize and get a measure of how efficiently a column can separate compounds independent of the length of the column. And to do that, we measure the height equivalent theoretical plates, which is defined as the length over the number of theoretical plates. And essentially, the smaller your HETP, the more efficiently your column is able to separate your compounds. So these are this provides a summary of how you process the peaks to calculate data about your column and calculate the retention time for your compound. Let's now take a moment and let's unpack the theory behind how gas chromatography actually functions. Okay, so once your sample is injected into the column, we have our column, which is typically consisting of a stationary phase, which is a compound bound to the column surface. As our sample passes through the column, it will condense and interact with the stationary phase. And this interaction is based on the intermolecular forces, column temperature, and the flow rate. The flow rate is a measure of how quickly your carrier gas is passing through your column. A high flow rate is pushing your sample quickly through the column, decreases your retention time, and may result in decreased resolution, as we're not able to establish this liquid vapor equilibrium as readily. Just like in a distillation, this liquid vapor equilibrium between our sample in the mobile phase and our sample in the stationary phase is critical for an effective separation. Now, if we start upping the temperature, the sample will be less likely to be absorbed to your stationary phase, to be deposited as a vapor on the column. And as a result, our sample will pass more quickly through the column. Now, high temperature is not always bad. High boiling compounds can give pretty broad peaks if the temperature is not high enough, as our sample sluggishly absorbs to the column as it progresses through the column. So sometimes we do have to increase the temperature in order to get a reasonable peak shape. And that's pretty easy. We've already optimized the oven temperature to maximize separations. Okay. So, different compounds pass through the column at different times based on their intermolecular forces. We can control the exact conditions of our column to optimize the separation. We can control the temperature, we can modify the stationary phase, we can control the flow rate. Okay, we've already optimized all of these background pieces of information in order for these separations that you're going to watch in the video to run as smoothly as possible. And I really can't understate that this optimization does take time. But we've done this optimization, and now we're ready to separate our compounds using preparative gas chromatography. So again, we're going to inject our mixtures into our gas chromatography column. We are going to observe each compound as it passes past our detector, and we're going to calculate the retention time, peak height, and peak area. Okay? Now that once we've calculated each of our peak heights and peak areas and we've collected each of our samples, then what we're going to do is we're going to calculate the recovered sample, which is the mass of our sample recovered from our gas chromatography column divided by the initial mass of our sample that we injected, which we can calculate using the density and volume of sample injected. This gives us our percent recovery for our sample. Now, in addition to calculating the percent recovery, we can also utilize our peak area to approximate and estimate the mass of sample recovered from this separation. So we know that the peak area is roughly proportional to the concentration or mass of sample. So if we process each of our peaks and we identify our peak height, retention time, and peak width, there are two main methods that are used to calculate peak area. The first is by using the width at half height times the height. So you take the height, you find the half height point, and you measure the width. The other method that we can use is we can physically cut out our peaks and weigh the peaks. The other formula that you can use for the area is by taking 1/2 the peak width times the height. This formula is also commonly used. Both of these methods are valid, and both of these methods will give relatively reproducible areas. I would like you to perform the area calculations using both of these methods and then compare your results. Now, once we have the peak area, the ratio of peak areas corresponds to the approximate ratio for the mass of each compound. So what I would like you to do is I would like you to calculate the peak area ratio and compare it to the mass ratio of our isolated compounds. And I'd like you to see whether the area ratio matches and agrees with or disagrees with our mass ratio. Does that make sense to everyone? Does that make sense to everyone so far? Does everyone understand what we're doing in this experiment? So, in the video, you're going to see a separation of a known mixture via preparative GC. You're going to be able to calculate and record from the provided data the physical properties and mass of our isolated material, and from the chromatograms which are posted on Canvas, you'll be able to calculate the retention time, peak width, peak area, number of theoretical plates, and height equivalent theoretical plates. And just as a reminder, you're going to do this for each peak. From this data, so derived from this data, you're then going to calculate the mass over mass ratio and the area over area ratio, as well as the percent recovery. For parts 2A through C, I've separated a series of 3 unknown mixtures, which are all found in your lab manual in experiment 2. And I provided you the refractive index and boiling point data. From the video and the chromatograms, you'll be able to calculate the following parameters. And with all of this data that you've collected, that you're going to collect from this experiment, what you're going to do is you're going to assign the identity of each component of each mixture, and then you're going to compare the physical properties to the physical properties of a pure sample. So you're going to need to make sure to watch the video very carefully to record the needed data. And then I'm going to want to see attached to your report a detailed analysis of your chromatograms. To show you an example of how you would analyze a chromatogram, allow me one moment to clear off this previous annotation. And what we're going to do is we're going to look at a sample chromatogram readout, and we're going to talk a little bit about how to process this data. This is exactly the kind of raw data that you would be processing in this lab. Okay, so here is our chromatogram. Okay, so first thing that we're going to do is we're going to assign our tangent lines. First thing that we're going to do is we're going to assign our tangent lines. So it's a point in our curve where the slope changes. In this case, the peaks are relatively well defined. So like this lead for the peak is almost perfectly aligned with the tangent line. So this first part looks quite good. Now for our second part, our tangent line, our point of inflection occurs around here. So we're going to draw our second tangent line. It doesn't have to be perfect as long as your method is consistent. Okay, so now what we're going to do is we're going to record the times when the two tangent lines intersect our x-axis. So in this case, we have T1 as about 5.8 and T2 is about 7.3, just as an estimation. We're then going to assign our peak height, and we're going to assign our retention time, which is the point when our two tangent lines intersect. So our retention time would be approximately 6.5. Is everyone following me so far in this analysis? Is everyone following me so far in this analysis? Can I get some responses in the chat that everyone understands what I'm doing in this analysis? We are looking and analyzing our peak to determine how efficiently our column can separate our compounds and to estimate the mass of each compound isolated. Okay, peak area is approximately equal to the mass. It's not exact. It's not exact, it's approximate. Okay, so now that we have the retention time, we know that we can calculate our width by taking T2 minus T1. So our width is here. We know that our height is about 6.5. So 1/2 height would be about 3.25, which looks like it's about here. So our peak width, we'd calculate our normal peak width to be 5.8 minus 7.3, which gives us 1.5 minutes. At 1/2 height, let's calculate the width at 1/2 height now. So the width at 1/2 height is 7.8 minus 6. Oh, sorry, 6.8 minus 6. It's closer to 6.9 minus 6, which gives us 0.9 minutes. Okay. So from this information for this peak here, we'll calculate the area using method 1 as 1/2 height times the width, which would be 1.5 minutes. And the area using method 2 would be the width at 1/2 height, which is 0.9 minutes, times the height, which is 6.5. So punching this into our calculator, that gives us our first area of 4.875, which in turn gets rounded to 4.9. And our second area turns into 5.9. Depending on the amount of precision you have for your peak, for your widths and the width at 1/2 height, you may get more or less accurate results. As long as your peak area methods are consistent for your comparison, you're more than good to go. We can also calculate the number of theoretical plates, which would be equal to 16 times the retention time over the peak width squared. So then for this peak, N would be 16 times our retention time of 6.5 over our peak width, which will use the overall peak width of 1.5 minutes squared, and that gives us a number of theoretical plates of roughly 300, which we'll write as 3.0 times 10 to the second. Gas chromatography columns are incredibly efficient at separating compounds, and as a result have a relatively large number of theoretical plates. If we wanted to calculate the height equivalent theoretical plates, we'll take the length in centimeters over the number of theoretical plates. So I'm going to leave to you the conversion from our 8-foot column into centimeters to calculate the height equivalent theoretical plates. Do these calculations make sense to everyone? Do these calculations make sense? Let's analyze the second peak just for completeness. So I'm going to quickly draw both of my tangent lines. So reading off, T1 is about 10.8. T2 is about 13.1. So our width is 13.1 minus 10.8, which in turn gives us 2.3. Our retention time is about 11.8. Reading off our height now. Our height is 6.0, so half height. Reading off our width at half height. Our width at 1/2 height would be equal to 12.5 minus 11.1. Well, probably closer to 12.4, which gives us 1.3. So now if we're calculating the area of this peak, so the area would be equal to 1/2 times the width times the height, and that in turn gives us an area of 6.9. looking at our area calculated using method 2, we have 1.3 times 6.0. And that gives us a second area calculated using our second method of 7.8. So now that we have each of our peak areas, what we can do is we can calculate our area ratio. So if I label this as peak 1 and peak 2, A1 over A2 would be calculated to be 4.9 over 6.9, which gives us an area ratio of 0.71. And I'd like you to compare this area ratio to the mass ratio of isolated compound. Does that make sense how to complete the calculations? Does that make sense to everyone? How to complete these calculations? So, as a summary, just as a summary, you're going to watch the video and you're going to collect data from the video. I've provided you the chromatograms corresponding to each experiment in the video. You're going to analyze and figure out the retention times. The retention times that I give you in this table are not exact. You're going to have to figure them out on your own from the chromatogram. I've also provided you the physical properties such as the refractive index and boiling point for the samples that I measured outside of the recording. The chromatograms for mixtures 1 through 3, which are your unknowns, are found in this PDF titled 2ABC. I'm going to need to see your analyzed PDFs attached to your report. And you can even include your analyzed PDFs in your results section. For your unknown mixtures, I provided you the retention times and physical property data, and your job is to assign every single component of each of these 2 mixtures to one of the 8 compounds found in your lab manual, experiment 2. I'm also expecting, as we've done here, a calculation of the peak area, peak width, retention time, number of theoretical plates, height equivalent theoretical plates, and the peak area and mass ratio. You can also print out the graphs when you work on them and process them. That's more than okay, but it should be clearly legible how you are processing your graph. And I need to be able to see your tangent lines and see your retention times assignments to make sure no major errors have occurred. So this is a 2-week experiment. So we're going to spend this week and next week working on this experiment. And just as a side note, we'll be giving a lecture next Thursday on Lab 3D, which utilizes the exact same method of analysis. So it's really important that you understand and are comfortable analyzing chromatograms and processing and calculating area percent, mass percent, and all of these other quantities as you'll be doing these types of calculations, not only for these gas chromatography labs, but also for the subsequent experiment 3D, which focuses on fractional distillation using a spinning band column. Okay, so I've summarized today's laboratory, and don't worry, we'll be doing a refresher next Tuesday as well, going over this lab again, talking about the theory and refreshing the calculations. But I wanted everyone to have this background so they can get a head start in the lab and start working on it now, because it is a 2-week lab and it takes a fair bit of time. I know it seems like I was able to process these spectra pretty quickly, but when you're working on it, it may take you a little bit more time to draw out all the lines and get these calculations sorted. So with that said, the remainder of this period will be open for you to work on watching the video, collecting the mass data, and analyzing the chromatograms for each of these compounds.